Hi everyone, welcome to Basic Science Series. I'm your host Lokinder Kumar, and today we will discuss staining profile as well as some of the medias that are required for the growth of Staphylococcus aureus. I hope that you have seen my previous video on Staphylococcus aureus where I have discussed all the important points regarding the Staphylococcus aureus. I have touched the base on those specific points. Here I'm going to elaborate some of the important points. For example, clump staining. I will also discuss the MSA profile. MSA profile is the Mannitol salt agar profile, and I'll also discuss the blood agar profile. As you can see, I have designed these illustrations that is going to help you to understand what I'm trying to show you, okay, and what we are trying to discuss. So I hope you're gonna like the video. If you like the video, then please do subscribe to the channel so that I can make more videos that's going to help you in your studies and your exams. So I hope that you're gonna stay tuned to the channel. With that note, let's start the video presentation. All right, with that note, let's start the presentation from slide 1. In the slide 1, as you can see, I'm trying to discuss the structure of Staphylococcus aureus. When you perform the gram staining profile, you will get this kind of profile for the Staphylococcus aureus. You will get clumps. You will get clumps of these circular bacterial cells. And the size and the diameter of this particular cell is 1 micrometer. You need to remember that. This is the size of, typical size of one Staphylococcus aureus bacterial cell, the circular cell. And when you stain them, you will see that they will be forming certain types of clumps. They will look like grapes. So this is how you can differentiate between Staphylococcus and Streptococcus, because Streptococcus, they will form chains, right? Let me use this red colored marker or highlighter so that I can direct you, right? So this is the diameter that I'm trying to show you. From one point to another point, it's 1 micrometer, and these are the clumps that you're going to see under the microscope, and probably you're going to use oil immersion lens and 100x magnification. As you can see here, I have shown you that it's going to stain gram-positive because it's a gram-positive bacteria. And we know that gram-positive cell wall is different than the gram-negative cell wall. That's why they stain differently, right? In my previous videos, I made presentations on gram-positive cell wall as well as gram-negative cell wall. So you can watch those videos and understand what is the composition of the cell wall. On the basis of that, they are differentially stained. And if we talk about the presence of the Staphylococcus in humans, then you will look for the nasal area. In the nasal passage, you will find Staphylococcus aureus in case of human. Now let's move on to the next slide. In the next slide, I am trying to show you the profile of Staphylococcus aureus colony on blood agar. Blood agar is a differential and the enriched media. Differential, why differential? Because on the blood agar, you can differentiate between different species of the same bacteria, and also enriched media because it's enriched in nutrients. Now you will find light to golden yellow colored colonies on blood agar for Staphylococcus aureus because it produces that particular pigment. And in case of Staphylococcus epidermidis, which is non-pathogenic bacteria of Staphylococcus genus, is going to form white colonies, and it's going to appear white because of the white pigment, right? Another important point, that this particular bacteria, that is Staphylococcus aureus, is going to form beta-hemolysis. Beta-hemolysis is different from the alpha hemolysis because it completely lies the RBCs around the colonies, and you can clearly see the zone. They are very transparent and clear because of the complete lysis. And in case of alpha, you will see some greenish colored zones around the colonies, right? And I can make a separate video on the types of hemolysis of different bacteria, and we can discuss those. But here you need to understand that Staphylococcus aureus forms beta hemolysis on blood agar, and the size of the colony is 1 to 4 millimeter in diameter based on the time of the growth and the inoculum that you are putting in the plate, right? And the size and the shape of the colony the colony, they have sharp border. So it's not a diffused border. As you can see, the border is very sharp for these colonies. And then you have round colonies. They are round in shape and also convex. Convex colonies. They are not flat, they are not concave, they are convex, typical for Staphylococcus aureus. Now let's move on to the next media, that is mannitol salt agar, MSA. It has, as the name indicates, it has mannitol, and it also has the high concentration of salt. Because Staphylococcus aureus can tolerate high salt concentration and can ferment mannitol, this is a differential and an enriched media for this particular organism. Now, the salt concentration varies from 5 to 7%, and in this particular media you'll find mannitol, right? And mannitol is going to get fermented by Staphylococcus aureus, and it's going to produce acid. And when there is acid production, the media, the color of the media will change and it will turn yellow. On the basis of that, you can maybe say that there is a chance that you have a Staphylococcus aureus in your mannitol salt agar plate, right? As I already mentioned, that you need further confirmation of this particular organism, and in my next video I'm going to discuss some of the important tests related to that. Now, why it changes the color? Because the media has phenol red, and that's the pH indicator, and when there is acid production there will be the change in the pH, right? And that change is going to change the color of this indicator, and because of the change in color you can easily differentiate between acid producing and non-acid producing bacteria. In this case, the colonies, they will have sharp border, they will round, and they will also produce the convex shaped colonies, and you can differentiate between pathogenic and non-pathogenic on mannitol salt agar based on the change in the media color, right? So Staphylococcus aureus, which is pathogenic, is going to produce small colonies and it's going to change the color of the media, but Staphylococcus epidermidis is not going to change the color of the media with those small colonies. And you can say that on the basis of the change in the media color, you have Staphylococcus aureus or you have Staphylococcus epidermidis, right? And now I have just discussed the basics, and I want you to study more about the mannitol salt agar. And if you have any questions, then please post your questions in the comment section. Now, let's move to the final slide, which is the same slide that I always use, which is the first slide, right? And as you can see, I have made this gram staining profile to just summarize what we have discussed. And we discussed that it's the gram-positive bacteria. The size of the bacteria is 1 micrometer in diameter, and colony size is 1 to 4 millimeter in diameter. And also you can see it's going to produce the colonies, and it's going to change the color of the mannitol salt agar if you have Staphylococcus aureus. And in the blood agar, you will see clear zone of hemolysis, which is beta hemolysis, and you can say Staphylococcus aureus is beta-hemolytic in nature. So these were some of the important points related to its gram staining, related to its media requirement, and there are so much more on this particular organism, and I want you to study those points. And if you have any questions, then please post your questions in the comment section, and I'll try my best to answer those questions, right? In the next video, I will bring catalase, coagulase, and we'll discuss the mechanism of those particular biochemical tests in detail. Okay, so with that note, I will just conclude my presentation, and if you like the video, please do not forget to subscribe the channel so that I can make more videos on similar topics like this. Okay, with that note, I'll just conclude my presentation, and I will meet you in my next video presentation. Thank you and take care.